I assure you that what you will observe is a vast wasteland.
Free and uncorrupted communication.
It's my great pleasure now to introduce our keynote speaker this afternoon, Professor Erkki Ruoslahti. And Professor Ruoslahti has recently moved his laboratory to UC Santa Barbara and joined the MCDB faculty with the title of Distinguished Professor while retaining his affiliation with the renowned Burnham Institute in La Jolla, California. And most of you may know Professor Ruoslahti is internationally recognized for his research in the area of cell adhesion, extracellular matrix biology, and tumor metastasis. He co-discovered the protein fibronectin, one of the best characterized ECM adhesion proteins, and his laboratory then proceeded to identify the major adhesive sequence within fibronectin, the now famous RGD peptide, which has subsequently led to the development of a variety of drugs to control things like blood clotting and angiogenesis. Dr. Ruoslahti's lab then went on to identify the cellular receptors for fibronectin, and with several other labs at the same time described the integrin family of receptors, which are now known to be crucial for cell adhesion, survival, and proliferation of a number of different cell types in the body. And Dr. Ruoslahti has authored over 420 scientific articles in these areas of research. A little bit about his background. Dr. Ruoslahti was trained at the University of Helsinki, earning bachelor's, MD, and PhD degrees there. And then after carrying out postdoctoral research at Caltech, he held professorial positions at the University of Helsinki and the University of Turku in Finland. But then he couldn't resist Southern California, I guess, and came back, and he moved back to the U.S. as a senior research scientist and then eventually director of immunobiology at the City of Hope in Duarte, California. And then he went to the Burnham Institute, which was then called the La Jolla Cancer Research Foundation in 1979, and he's been there ever since, serving as scientific director, vice president, and eventually CEO from 1989 to 2001. And I don't know if you've ever been down to the Burnham Institute in beautiful La Jolla, but it's really grown quite a bit since its beginnings. Under Dr. Ruoslahti's leadership, the Burnham grew from basically a single building with a handful of employees to a campus of 10 buildings with more than 500 scientists working on everything from diabetes to neurological conditions to heart disease. And in fact, there's even a street named after Dr. Ruoslahti on the Burnham campus. So, Dr. Ruoslahti has served on numerous government review panels, editorial boards, and scientific advisory boards for both academic institutions and biotechnology companies. And if I listed them all, he wouldn't have a chance to speak today because it would take too long. He's also been active in establishing several companies based on his own research. His honors include elected membership to the U.S. National Academy of Sciences, the American Academy of Arts and Sciences, and the European Molecular Biology Organization. And he's the recipient of many prizes and awards, including the Japan Prize in 2005. And in his home country of Finland, he has been knighted, and he is a Knight of the Order of the White Rose of Finland. His current, you can't say that about every speaker that comes up here. His current research is at the interface of nanotechnology and cancer biology, with emphasis on identifying molecular addresses that are found in tumors in the vasculature of various types of tumors. And this has great potential for the development of new cancer therapies. The title of his talk today is Vascular Zip Codes in Drug and Nano Device Targeting. Erkki.
Thank you very much, Dennis. It's indeed an honor to be giving this lecture for a newcomer like me, as you see. I'm in the process of moving my laboratory to the molecular biology department, and it'll still be affiliated also with the Burnham Institute. And on the background here is one of the attractions that brought me here. And here's another one. As well as the names of some of the people who are involved in the work that I'm going to talk to you about. Our lab is beginning to get going here in Santa Barbara, but a larger portion of it is still in La Jolla. I'm particularly going to talk about work that was done by Dmitry Simberg with nanoparticles and peptides that were isolated by Tero Järvinen and Jan Pilch and Liangin Zhang among the current members. And you can see I'll mention the other names as we go along. I should point out that I have quite a few collaborations that are quite important. I'm going to tell you about the collaboration between Sangeeta Bhatia, Mike Saylor, and myself. And I share 2 grants with already with people here at UCSB, Matt Tyrell and Patrick Doherty. So we have great expectations for this particular aspect of the work. So what I'm going to tell you about is vascular heterogeneity. And there's so much vascular heterogeneity that I think we're justified in talking about vascular zip codes, a whole system of heterogeneity. And the importance of being able to target the vasculature specifically is that it is accessible. And unlike, say, tumor cells, which are genetically quite malleable and tend to evade treatments, the vasculature, even in tumors, consists of normal cells, and they don't have the capacity to change in response to treatments. And so I'll tell you about that vascular heterogeneity, particularly in the context of targeting of nanoparticles into tumors. And the promise of using a nanoparticle in therapy is that when you take a drug, it can only do so much, whereas a nanoparticle you can engineer to have multiple functions. And that's the approach. And we take a modular approach to this, such that we work on one particular function at a time. But as far as using nanoparticles in tumor imaging and therapy, the hope is to make them go into the tumor, release a payload, send a signal out, and so forth. And what that requires is being able to first physically target the particles to where we want them. And that's my specialty, in particular in the nanotechnology projects that we're doing. Now, I'll tell you how we can target either the inner lining of the blood vessels, the endothelial cells. We can target tumor cells themselves, and even the extracellular matrix in tumor vasculature and in tumors is special. Now, there are a number of hurdles to work on before nanoparticles can be targeted readily into tumors. And one of them is that they don't penetrate tissue well, as I'll show you. And they tend to be taken up by the reticuloendothelial system, which is a system of phagocytes in the liver and in the spleen. and lymph nodes. And that's a big problem in applying nanotechnology in vivo. I already mentioned these particular goals. So we've taken two particular aspects of this list here to work on initially. Avoiding the RES and amplifying targeting. So the way we find the vascular zip code addresses that we use in targeting is that we use a nanoparticle to probe the vasculature. And that nanoparticle is a virus that infects bacteria, Phage. And you can take bacteriophage and engineer onto the surface a peptide or a piece of protein or an antibody in such a way that you form what is called a library, a huge collection of different peptides, such that every phage particle expresses its own peptide. And you can now take this kind of a library and isolate from it what you're interested in, which in our case is that the phage would home to a particular organ or diseased tissue in the body. And you can then use the phage's genome to figure out which peptide it is that made the phage go to the target. And our innovation really here was that we did it in vivo in live mice. So we inject a library with 1 billion or so peptides into a mouse, collect the organ that we're interested in, rescue the phage from it, and then by repeating the process, end up what we call homing peptides. Peptides that have a specific affinity for our target. And we won't go into that. That's just an enrichment step that we have added to the process later on. So using this technology then, we've been able to show that essentially every tissue puts a signature on its vasculature. And why I say that is that every tissue we have taken so far has proven to have its own vascular zip code, such that that tissue, let's say the brain, its vasculature contains molecules that are not expressed anywhere else in the body, and so on through that list. And I have one example, a recent one here, where we isolated heart homing peptides. And you can see that when we have the phage home after an IV injection, we get a tremendous enhancement of the accumulation of the phage at the target relative to other tissues. So every tissue, normal tissue, puts a signature on its vasculature. Well, our particular interest is tumor vasculature. There are two kinds of vessels in tumors. There are blood vessels that bring in blood and take it out into the venous circulation. And these blood vessels are necessary for the growth of the tumor, in that as the tumor grows, the vasculature has to grow with it. Otherwise, the tumor is starved. And starving tumors in this manner is a very popular new therapy that was just the first product, an antibody against a growth factor that stimulates tumor angiogenesis, was approved for human treatment. Angiogenic blood vessels are different from normal blood vessels, and I'll show you some examples of that. The other circulation, which has only an afferent branch to it, is the lymphatic vessels. They're also important in tumor, not for the tumor growth, for the growth of tumors, but as a conduit of metastasis. And as I'll show you, the lymphatic vessels are also special in tumors, and we can target them. Now, I don't want you to try to absorb all this information here. I just want to make the point that we have many, many peptides that specifically target tumor vessels. Some of them target the blood vessels, some target the lymphatic vessels, some target the extracellular matrix of the vessels and the tumors themselves. And then one group of peptides that we've studied recently target fibrin deposits, clotted plasma proteins in tumors. Turns out that tumors, but not normal tissues, have clotted plasma proteins in them, and those can be targeted. So let's look at some examples. Here's an example of a peptide that targets tumor vessels, and it targets vessels because they're growing vessels, angiogenic vessels. Typically, when we identify a peptide with the phage display method that I explained to you, we get rid of the phage, we synthesize the peptide, and then label it so we can follow it. And here's this peptide we call F3 injected into a mouse that carried a tumor. And what you see the F3 peptide doing is it accumulates in the nuclei of the blood vessel endothelial cells. Here's one labeled with the red dye. And then the peptide also gets into the nuclei of the tumor cells themselves. In fact, this peptide, and I won't go into the details here, is a cell type specific internalizing peptide. There are peptides that are used a lot now to get things into cells, but those peptides are non-specific. They make whatever payload you choose to go to every cell. Our peptides take the payload only to the tumor vessel endothelial cells or tumor cells themselves. And I won't go into more detail about that. Here's another example of a peptide that is particularly effective in spreading into the tumor. So here you see the blood vessels. After several hours, although the peptide starts with the blood vessels, it has spread like this. All the green color here is the peptide. You can imagine what could be accomplished if this green colored substance, fluorescein, would be a drug instead of just a colored substance. The differences between normal and tumor vasculature are very subtle, so subtle that, that they even extend to pre-malignant lesions. Here's a tumor system that Doug Hanahan at UCSF established, and we collaborated with them to ask the question, would we be able to tell apart the blood vessels of pre-malignant lesions from normal and from tumor? And the answer is yes, we can. We have a set of peptides that in this system recognize the pre-malignant lesions called here angiogenic islets, but not normal islets in the pancreas and not full-blown tumor in the same system. We have other peptides that recognize tumor vasculature, but not normal or pre-malignant. And then the third set that recognize shared epitopes in both of those. So the changes in the tumor vasculature actually follow the development of the tumor. And they can also be tumor type specific, something I'm not going to show you today, but that is something we've also found. Tumor lymphatics are different from normal lymphatics. Here's a peptide we call LIP1. And when we inject LIP1 into a tumor-bearing mouse, the peptide ends up in the nuclei of the lymphatic vessels. Here we used a lymphatic marker called podoplanin. And the peptide goes to cells that clearly form a vessel and stain with that marker. And then the peptide later on spreads into the tumor. So here's another cell type specific cell penetrating peptide, now recognizing tumor lymphatics. We've done more with tumor lymphatics since we published that work that is just about to come out in Cancer Research and done by Lianglin Zhang. Now, Pirjo Laakkonen, who did the LIP1 work, already noticed that LIP1 is cell type specific. It recognizes breast cancer lymphatics, but not lymphatics in this melanoma. So Lianglin took on the task to find lymphatic homing peptides for the melanoma, and he found this peptide. We name often our peptides based on the first 3 amino acids in the peptide, and somewhat unfortunately they turned out to be LSD in this case. Anyway, we think this is real regardless. And this peptide has the exact opposite specificity to LIP1 and also recognizes lymphatic vessels. So there's a whole zip code system in the lymphatics as well as in the blood vessels. So some of the things one can do with these peptides, then I already alluded to. We haven't done much with our new peptides. That's ongoing work that I will not talk about yet. But our first generation peptides, that one of which includes this RGD sequence that Dr. Clegg mentioned, that we discovered many years ago. And these RGD peptides home to tumor vasculature because 2 of the integrins that we, particularly alpha V beta 3, which we also found in the 80s, are upregulated in tumor vasculature. So peptides that contain the RGD home to tumors because they bind to this integrin. So we used peptides like this to target doxorubicin and also this peptide, which is pro-apoptotic, meaning that if it gets into cells, it opens up their mitochondria and causes apoptosis. And the question was, can we target this peptide into tumors so that it would kill tumor cells? And indeed it does, and we published it several years ago. The tumor growth is inhibited when we treat tumor mice with this particular combination. Now, we can also take this type of a peptide and suppress angiogenesis in arthritic synovium. And we can take a different peptide that binds to the normal prostate vasculature, and we can destroy the prostate of the mouse. The point of this is that we can take this essentially non-specific compound, and depending on what targeting element we put it, we get it to go into different places. We've also lately used one of the LSD peptide in a similar manner, and here's the result. It both kills lymphatic vessels in the tumor as well as suppresses tumor growth. And we believe that the suppression of tumor growth is because the target for this peptide is shared by the lymphatic vessels and by the tumor cells. Now, some of our peptides are extraordinarily effective in accumulating in tumors. Here's again the LIP1 peptide I already mentioned. Here you see a mouse outlined by the autofluorescence of its fur. or skin rather here. This is a nude mouse. And here's the tumor 20 hours after we injected LIP1 labeled with fluorescein into this mouse. If we inject a control peptide similarly labeled, the tumors have absolutely no fluorescence we can detect. And note that we can detect this fluorescence just on a regular light table. No computer analysis needed. You can just see the tumor glow. And here's the difference between the tumor and various normal tissues in the tumor mouse. So quite impressive targeting. The extracellular matrix, as I mentioned, is also different in tumors. Here's an example of a peptide that homes to clotted plasma proteins, which are in the matrix within the tumor. Also a very effective peptide. And then, as I mentioned earlier, we target nanoparticles with these peptides into tumors. We started out with quantum dots, which are highly fluorescent nanoparticles under 10 nanometers in diameter, and showed that we could get exquisitely specific targeting to blood vessels if we use the blood vessel targeting peptide, to lymphatic vessels if we use the lymphatic targeting peptide, or in this case into the normal lung vasculature with yet another peptide. We sent this paper several years ago to Nature, and they didn't even review it. And we asked Nature Biotechnology whether they would review and perhaps accept it. They didn't take it either. Neither did Nature Materials. So I sent it to PNAS because I thought it was a pretty nice story. When it came out in PNAS, Nature Materials wrote an article that was longer than our original article on it, which was picked up by Nature Biotechnology and Nature. But quantum dots don't really. That was a good demonstration that this can be done, and it hadn't been really done before in vivo. But the quantum dots don't really do that much. So we have lately switched to iron oxide, where one can detect a signal with MRI and even hope to control them with MRI, perhaps from outside the body. And we again put the homing peptide on them, and then are able to target them, and I'll show you some examples in a moment. I mentioned in the beginning that other than targeting, one of our goals is to accomplish self-assembly of the particles. And the idea here is the same as what platelets do in blood clotting. When one platelet finds a breach in the vascular wall, it gets activated and it calls in more platelets through aggregation. Or if you want, you can think of if you forget a drop of dog food on the kitchen floor, one ant finds it, and pretty soon there's going to be 1 million ants there. So that's what we wanted to accomplish. And we have two systems that I'm just briefly going to describe to you that do work in this manner. One of them was designed in Sangeeta Bhattia's lab with some minor help from us. Just came out in Angewandte Chemie and made actually the cover of the journal. And it's based on the following idea. There's a receptor on the surface of the iron oxide particles that is covered by a chain of polyethylene glycol attached to the receptor by a protease-sensitive linker. Now, when the protease in the tumor cleaves off the polyethylene glycol, the particles will aggregate because some of them have the receptor and some have its ligand. And this actually works in vitro, and I think I'm not going to explain the details of it, just a demonstration that it does work. If the aggregation of the particles is triggered, they become attracted to a magnet, whereas if they are individual particles, they don't. And here in this tube, extract, or actually culture media, from tumor cells was added, and here it was inhibited by an inhibitor of the protease, and you can see that here we see aggregation and not in that tube. And you can bring these particles onto cells as aggregates like this, and there is the control. So this system clearly functions very nicely in vitro, and we're just about to take it in vivo. Now we have also a system that we mostly designed by doing in vivo experiments, and it indeed works in vivo. And it was a reiterative process in that we would design something in vitro, test it in mice, and more often than not, it would completely fail. We'd go back and redesign, and in this manner, we've actually now made the system work. And I'll tell you how. But first, here's the problem. I showed you very impressive tumor homing of fluorescently labeled peptides. But if you take a nanoparticle and you inject it, you don't get impressive tumor homing. You get impressive liver homing, spleen homing, and lymphatic node homing, because those organs contain the reticuloendothelial system that takes up your particles, and very little goes into the tumor. Here's a comparison of a fluorescent peptide. Here's the mouse's tumor, and here's its bladder. The extra peptide gets excreted into the urine, and what accumulates in the tumor lights up the tumor. A huge contrast in the pharmacokinetics between those two systems. So our task then became to prevent the reticuloendothelial cell uptake. And the way we have done it so far is to use decoy particles. And Dmitry Simberg, who did this work mostly, he noticed that the proteins that bound to the particles that ended up in the liver tended to carry proteins that were clearly metal-binding proteins. So we used nickel liposomes as the decoy particles to take away those proteins that otherwise would bind to our targeted particles. And indeed, it greatly improves the tumor homing. And it greatly improves the blood half-life of the particles, as shown here. After the decoy particle treatment of the mouse, our targeted particles stay in the circulation much longer than they would otherwise, and apparently because of that, a lot more ends up in the tumor. So here's the amplification then, that we didn't really design. It just happened because we got so much iron oxide into the tumor vessels that it actually apparently elicits blood clotting in the tumor vessels and obstructs the vessels as a result. Here's a demonstration of how the blood vessels are full of this amorphous material, and that there's so much iron oxide in this tumor vessel that we can actually see it in light microscopy. And the demonstration that there is clotting in the tumor vessels comes from staining for fibrin, the plasma protein that primarily makes a blood clot. And I'm not showing to you the data, but we can inhibit the clotting by treating the mouse with inhibitors of blood clotting. And this clotting takes place in vivo. We have looked at the mice while it happens with what is called intravital microscopy, where you can actually observe what is happening to the tumor in a live mouse. And we can see these clots forming in blood vessels as we observe them after the nanoparticle injection. Now we start getting pretty impressive tumor homing. Here's an example of tumor imaging. Here's the tumor as compared to a control injection where the particles were labeled but not targeted. This peptide is one of our favorite tumor targeting peptides. Now, this is kind of complicated, but it's our best demonstration to the effect that we are actually getting amplified targeting. What we did here was we would inject the mice with targeted, non-labeled particles. And of course, in that case, we'd not see the particles in the tumor because they were not targeted. If we then would add the non-targeted particles or inject them together with here the particles were non-targeted and labeled. If we then would take the non-targeted labeled particles and inject them together with targeted unlabeled particles, we'd see the tumor because these untargeted particles accumulate in the tumor. I told you it was complicated. I got mixed up myself there, but this is our best demonstration that we're actually achieving amplification as we had planned to and hoped to achieve in this system. There's one other promising thing that has come out of the system that we're not, it requires more work, but it's so intriguing that I thought I'd show it to you at this point. And that is that we seem to get necrosis in the tumor when we inject those particles that cause clotting in tumor vessels. And it makes sense because the tumor needs blood circulation to stay alive. If we clot the tumor vessels, we should see reduction of necrosis short-term in the tumor. And we do, although we don't have enough mice to be sure that that's really a causal relationship. But it is intriguing. So what we have here is a system where we've built in an amplification to the system of the particle accumulation in the tumor. We get very strong signals from the tumor, and we've used fluorescent labels so far, but of course we can go to MRI, and we may even get a treatment effect as a result. So what I've shown you is that the blood and lymphatic vessels are different in such a way that there is what we call a zip code system of molecular targets there. Tumor vessels differ from normal vessels. The differences are related to the growth of tumor vessels, angiogenesis. Some of them are tumor type related or tumor stage related, as I showed you. Tumor lymphatics differ from normal lymphatics, providing another target. We've so far only found tumor type related and tumor stage related differences, but there probably are lymph angiogenesis related differences as well. We just haven't hit upon any of those yet. The vascular and tumor extracellular matrix is different from normal matrix. There are clotted plasma proteins in tumors, and ECM proteins, something I didn't show you, but that we're actively working on. The markers of tumor vessels enable new therapies, guided drugs, as I showed you. They also, something I didn't get much into, the targets of the peptides, many of which we have identified, provide new druggable targets. And finally, then, the vascular zip code molecules allow one to target nanoparticles into tumors, and that is our main interest at the moment. Thanks very much.
Questions? Any questions?
I have one short question and one more general question. One of which is, how big is your superprimagnetic ion particle size?
Well, we mostly use about 50 particles that are about 50 nanometers in diameter.
And how about since many of these fields are already being used in human for molecular imaging in human?
Yes.
What is your actually plan in utilizing homing peptides along with these magnetic particles in human study? I think that will be. Do you have any plan for doing such?
Well, absolutely. That's where we're headed. As I mentioned, I showed you fluorescence results because it's so much easier to. A mouse is completely transparent to fluorescent imaging, but you couldn't do that in humans. But we're working on iron oxide, and as you mentioned, dumb iron oxide particles, non-targeted particles, are already being used in the clinic for imaging. Now we hope to be able to make major strides in making them more specific and making smaller tumors visible and new things visible that can't be imaged now. Yes?
I wasn't clear about how the amplification occurs. Does it bind to the particles that are already accumulated there? I mean, is that how the amplification occurs?
Well, I glossed over that because we don't fully understand it, but we think that it's actually a trapping thing, that because the particles are circulating while the clotting occurs, that it just traps the extra particles into that otherwise wouldn't get into the tumor or wouldn't stay there. But there may be more specificity to it. That's something we're studying right now.
In the back, yeah.
Thank you. Can you use signs of changes in the vascular and lymphatic systems to provide earlier diagnosis of tumors?
Well, that again is what we hope to do, and as I showed you, we can even detect vascular changes already in pre-malignant lesions, and depending on which peptide we pick, we can separate, we can tell apart pre-malignant from malignant, as I showed you, at least in one system so far. So I think that potential definitely is there. It just to get anything from this stage to the clinic, that's a long process, but we hope to start the first steps in that process fairly soon. Yes, sir.
Do you know, if any idea, how easy it's going to be to transfer those homing peptides from a mouse system to humans?
Yes, good question. I don't think it is going to be a problem for the following reason. Peptides bind to conserved sites in proteins. Unlike antibodies, because when you make an antibody, immunological tolerance plays a role in it, and antibodies tend to favor sites that are not conserved. Peptides favor sites that are conserved. For example, the RGD system, it works the same from Drosophila to humans. And every time we've had a chance to test our peptide in humans, we've seen the same thing. Sometimes we're able to do that with overlay. We can take sections of human tumors and overlay them with either phage or the peptide and see the specificity and binding. And so it doesn't seem to be a problem. On the other hand, of course, for nanoparticle targeting, the peptides may well be the ultimate tool, because you're dealing with multiple multivalent binding, and even if the affinity isn't that high, or even if some of the peptides become proteolytically eliminated, there's another 1,000 to do the binding left. Now, in some other uses of these targets, I see this going so that we identify a peptide, then we identify its target molecule, and then figure out whether that behaves the same way in the human, and then use that human target molecule to find a better peptide, a small molecule, an antibody, aptamer, whatever. And we've already done this process in some cases, switching from the peptide to an antibody. Yes. Oh, yes, sir. Sorry.
Can you guide and focus the nanoparticles externally? For example, if it is an intracranial tumor with transcranial magnetic stimulation, for example, of certain frequency?
Yes, that has been done, and there's actually a very clever system that is suitable for local treatment, and that is to use nanoparticles that have a gold shell on them. And you can then use RF or laser, I'm not sure just exactly what they use for, literally melting the gold, and that generates heat in the tumor, and it becomes what is called hyperthermia. And that seems to work well. The problem, of course, is that you have to know where the tumor is. There are all kinds of ways of getting at tumors that you know where they are. The real challenge is to find them in wherever they are in the body.
But that would help, for example, with transcranial magnetic stimulation, because it depends on the frequencies, frequency dependent. So
Yes.
back
Yes. The other situation where local treatment might be useful is ovarian cancer, because it's essentially confined to the peritoneal cavity. And we've some designs in that regard. Yes, sir.
Have you looked at whether you can target cells in the circulation, such as CD4 cells? Obviously, knowing that some of the current concerns in HIV are after the treatment, the latency, one could target the cells and get in with either enzymes or synthetic nucleotides to eliminate the residual infection that's latent.
Yes, I'm sure that could be done. I'm not sure that our technology is the best way of accomplishing that, because once you know the target, you can take an antibody, and an antibody will not have a penetration problem because the cells are in the circulation. So I don't think our technology is suitable for that. Our strength is to get at the blood vessels.
Okay, we better move on. Let's thank Erkki for a nice presentation.